Hi everyone, welcome to my channel Pure Biology, and today I come with another video, that is the ABSISIC acid signaling pathway. This is the second part of the ABSISIC acid signaling pathway, and I have explained the different factors that are involved this signaling pathway in my first part. So you just check out the first part and then you come to the second part. Okay, and here I just try to explain the processing of signaling pathway. So this is the plant cell, okay, and this is the cell membrane, PM, plasma membrane, and this is the ABA hormone. So this is cytoplasm, and this is nucleus, and this is plastid. So what is happening here? When ABA binds with this GTG, this is the receptor, and this GTG is the member of the family GPCR receptor. And here the conversion of the GTP into GDP is done, okay, or is occurring. And the GTP is getting from where? This is attached with this GPA and GPA1. This from GPA1 GTP comes and then it is converted into the GDP. Clear? Now from here the one signaling coming, that is due to the addition of ABA to the GTG, signaling coming to the TFS, means the transcription factors, and these transcription factors become activated, and after the activation of this, the gene expression is occurring. Clear? And first one is the what is this? ABA regulated gene. Okay, this one is the ABA regulated gene. This become expressed, and the second gene is the plastid protein related genes. Okay, and then second part, what is happening when ABA binds with this receptor PYR, PYL, and RCAR. These are the receptors and members of the receptors. This is the pyrrolactin receptor. So here the PP2C, this is the protein phosphatase, that becomes activated, and due to the activation of this SNRK2S, this becomes activated. Clear? And due to the activation of this, the TFS means transcription factors, this also activated, and gene expressions start to occurring, or gene expression is seen. And in this part here is the channel means here this is the which one is this? This is the calcium related channel. Okay, calcium related channel, and here the calcium dependency and independency is important for the activation of sometimes ABA process, ABA signaling pathway. And here the important receptor is that CHLH. Okay, CHLH receptor. Okay, this is the CHLH receptor. Okay, so CHLH receptor, and this receptor when ABA binds with this CHLH receptor, and CHLH generally present in the plastid. Okay, so what is happening here? This proto 9, this become activated, and then Mg proto 9, this becomes activated, and then it is just chlorophylls that also that present in here plastid. You know all that the plastid, three types of plastid, chloroplastid. So this in the chloroplastid, this is happening. And when this Mg proto 9 that becomes activated, what is happening? This goes towards the nucleus. Okay, the signals goes to the nucleus to activate the plastid protein genes. So here two important gene expression is there in the nucleus. One is the ABA regulated gene, and another is the plastid protein regulated gene. So when the plastid proteins in the chloroplast, the proteins when activated, then these genes that is present in the nucleus related to the plastid, that becomes activated. But when this SNRK2S or this ABA generally activates the ABA regulated gene expression. Clear? So So this is the process signaling pathway, and this is the channel of calcium influx or outflux that is occurring. If calcium dependent process is seen, so in that case the calcium entry or influx is seen. If it is not calcium independent, then the outflux of the calcium is happening. So here the PP2C is the protein phosphatase that have to interact directly with this receptor. Okay, you can see that. And ABA dependent interaction that represses this PP2C phosphatase activity, which leads to de-repression of the events. So here important thing is that when ABA, okay, first PP2C that already bound with this PYR, PYL, RCAR. Okay, so here when ABA binds with this, its activity, PP2A's activity is totally not, one thing is that. Okay, so here the one thing is that when this ABA binds with this receptor, the phosphorylation activity, that is phosphatase activity of this protein, that is totally stopped. Okay, phosphatase activity stopped. So by the stopping this phosphatase activity, what is happening? This ACIN RK2 its activity that becomes rise, and that's why this TFS, that the transcriptional factors that becomes induced for the transcription of the signaling pathway continuing, for the signaling pathway continuing. So here this is written that ABA generally it stops the phosphatase activity. Okay, it stops the phosphatase activity because that in that way de-repression events. That means the activation of SNRKase that is occurring, and this activation of SNRK2ase, it's and another two classes of protein that phosphorylates that phosphorylates the serine and threonine phosphorylation. Okay, sorry, this is not phosphatases. Okay, there is wrong thing. I just have explained to you that SNRK2ase and PP2CS and PP2A, these are the phosphatases that regulate this serine and threonine phosphorylation. Okay, serine and threonine phosphorylation, and the activation of numerous regulators that are also involved in this ABA signaling process that occurs, or they become activated, generally the transcriptional factors. So PP2C that integrate generally its function is what? It integrates the information from the receptors and the secondary messengers to modify the functions to downstream pathway. Its function is that PP2C. So this is the abscisic acid signaling pathway, briefly explained here. I think you understood it properly. So in the next video I will try to explain another thing. Just continue with my channel, and if you are the newcomer, so obviously share and subscribe this channel. Thank you.